Thank you for the invitation to come here and speak today. And given the audience and the setting, I have decided to skip the usual 5 slides to introduce the area. I assume most of you will be familiar with the clinical perspectives and the history behind that. So in a second, I will sort of dive directly into the details. Just to give you an idea about what I will be speaking about today. And now this doesn't work. Okay, I will try to use the mouse instead then. Yeah, so what I will be speaking about is, I'll give you 3 stories that deals with the development of different ligands from different applications. It's about the imaging of 2A receptor, the activation of 2A receptor, and also a compound with a potential therapeutic application. I will not dive into the details. I'll sort of skip over and try and give you an idea about how we do things. I will also tell you a story where we failed miserably, just to make sure that you don't think it's always as easy as it might seem there. But let's start out with imaging of the 5-HT2A receptor. So this started when I involved in the CIMBI Center, the Center for Integrated Molecular Brain Imaging, which was established in Copenhagen something like 20 years ago. And one of the goals we set ourselves was to develop an agonist PET tracer for the 5-HT2A. And you might ask yourself, why do that? One of the ideas or thinking behind it was that we'd like to have a PET tracer that would be susceptible to endogenous serotonin release. So if you did an investigation in a patient or perhaps a pharmacological intervention that caused the release of serotonin, it would be cool if we would be able to monitor that and to quantify that. And then you could pick, in principle, any serotonin receptor to do that with, but given that the 2A receptor is one of the most widely expressed receptors in the brain, that seemed like a good idea. At the time, there was also developed PET tracers that were antagonists, but thought as well, or the thinking was if we were to develop an agonist PET tracer, that would, in principle, be more susceptible to serotonin release. In the end, that perhaps turned out not to be the case, but of course we didn't know that at the time. So, and what is PET? Well, that's an abbreviation for positron emission tomography, and just a brief, that means that if you have a positron emitting isotope, and once that emits or that decays, and then it sends out these 2 photons, and if you have them that inside a scanner, you can then detect a blip there and a blip there, and then you know that decay happened somewhere along the line of that one. And if you have a lot of these, millions of these, wherever these lines intersect, a decay happened. If you then introduce this isotope onto a molecule that binds to some biological target, then you will be able to monitor, yes, where did that decay happen? Well, that was where that compound was, and if that compound at that time was binding to a particular receptor, well, there's a good reason to believe that was actually sitting at that receptor. So if something is going to work as a PET ligand, so it needs to be able to do a lot of things, and I've just listed a few things here. It needs very high affinity and selectivity for the particular receptor you're interested in, and that is in this case the 2A receptor. And the affinity, it needs to be able to bind to that receptor, and also needs selectivity. It shouldn't bind to everything else, because then the entire brain will just light up. You should, of course, it should be able to label them and introduce these isotopes that actually decay. It should go into the brain rapidly. Makes perfect sense. That's where the target is we're going to image. You should have something called non-specific bind. That means, we want it to bind to the target, but not to everything else, because that's going to be our window of detection. We also need suitable receptor kinetics. That's something about how fast does it bind to the receptor or go off again. And also we would like some of these, if there are any radioactive metabolites, not to go into the brain. And this sounds all very fuzzy, but during this lecture, in the next 5 minutes or so, I'll give you, examples of how do we actually look at this and, deselect and select compounds. And also to do this, well, you need a compound. So you need a compound that binds to the receptor you're studying. And with respect to the 2A receptor, the usual suspects, LSD and Psilocybin and mescaline, seems like a good place to start. But if you then also look at sort of their in vitro pharmacological profile, what do these compounds actually bind to? Well, LSD and also psilocin, I'm sure you know the active metabolite of psilocybin, is a fairly promiscuous molecule. Also, given that the structure of psilocin is very close to that of serotonin, it's not surprising that it binds to if not all, many of the serotonin receptors, and also LSD is a very promiscuous molecule, also interacts potently with the dopamine receptors. So if you were to label something like LSD or psilocybin, you will not be able to see any selective interaction with a particular receptor. Mescaline is, from the starting point, a much more selective molecule. It primarily interacts with the 5-HT2A receptors, so that could be a good starting point. Unfortunately, it's not a very potent compound. So it's nowhere near potent enough to be able to do that. But unfortunately, back in the days, Shulgin and others, have optimized the structure of the mescaline, arrived at the 2CX family of compounds, here exemplified by 2GI, which is a much more potent compound. So it binds to the 2A receptor with a much higher affinity. And also some 20 years ago, initially Ralph Heim and then later Nichols, began to investigate the N-bomb class of compounds, here exemplified by 25,5 N-bomb, which again is an even more potent compound. And that was perfect timing for us, because we started looking into this and went through the literature to figure out what kind of compounds could we use for this. Well, if you look at the first two ones, very high affinity and selectivity for the 2A receptor, well, the affinity was there. Selectivity was perhaps a little bit dubious, but we thought we could work on that. And then there's the possibility to introduce the radiolabel we need. So in this case here, there's 3 positions in this N-bomb class where we could introduce this radiolabel. So that seemed like sort of the perfect starting point for this. So what we did was to take a number of representatives from this class and then introduce this radiolabel and then evaluated those as PET ligands. And what we got out of that was these curves here that is done in pigs. And just to walk you through one of these, if you take the bottom one here, so over here on the x-axis we have time, and up here you have brain uptake. Then you have 2 areas in the brain that is listed on the blue one, which is cortex, and the red one is cerebellum. We know that there's a lot of 2A receptors in the cortex, not that much, if any, in the cerebellum, so we would like to have high uptake in the blue line and not a lot in the red line. We would also like to have rapid brain uptake, so the higher up this curve goes, the better. That indicates that the more of the compound goes into the brain. We would like to have low non-specific binding. That is, again, the distance between these two. And then with respect to the suitable receptor kinetics, that means how flat is this? If it's a very flat line, that means that the compound is very sticky. And if we want to be able to displace the radioligand with something else, it should be something that readily dissociates. So the steeper this curve is, the more, in principle, easy it will be to displace this ligand. So going through these, and we then focus into 2 of these and try and look at these parameters, you can see there's a very big difference in the brain uptake between the 2. It's much higher over here. If you look at how flat these lines are, so this is a very sticky ligand. It's not easy to displace it, whereas in this case here, you have a much steeper curve. And also the separation between the blue and the red line is much better over here. And I would say, could you have predicted that? Well, if you look at the structures, it's a very small modification. It's only an exchange of an iodine for a bromine. So this was very much a trial and error sort of approach, but this compound here, the more we looked at it, the better it looked. And to fast forward, in a couple of years, then Gitte Moos Knudsen and her team at NIU then advanced this compound into humans in order to try and, can we use this as a tool to investigate binding, the distribution and the binding of ligands to the 5-HT2A receptor? And the answer is yes. So this you see here is a picture of a human brain. The participant has had Symbiose-36 injected in its carbon-11 labeled form. And you can see on this heat map that the white and the red, that indicates where the ligand is binding. And then you might think, okay, is this just non-specific binding to brain tissue? No, it's not, because it's possible to displace this with ketanserin, which is a 5-HT2A antagonist. So the difference between these 2 here, that is specific binding to the 5-HT2A receptor. So subsequently, Gitte and the rest of her team have done some cool things with this PET ligand, and they continue to do. A recent study, now it's a couple of years ago, was they did a study where they used Symbiose-36 and then took in patients and dosed them up with psilocybin. And then they looked at the more psilocybin you give to people, the more psilocin is produced. And if you also then monitor how much psilocin do they have in their blood plasma, then you can also see the reduced binding of Symbiose-36 in the brain. So you basically do a binding assay in the brain of living human beings. So further studies are ongoing with similar things like this, but again, it turned out that this was a very useful tool in investigating the distribution, of course, and also changes in expression levels, etc., of this in live humans. So imaging, that is primarily a game of affinity. So I said at the beginning that there can be a difference between agonist and antagonist radioligands. So agonist compounds that are able to activate the GPCR, in this case, elicit a pharmacological response, and an antagonist, something that just binds to the receptor and shuts it down. So in principle, you can image a target with both an agonist and antagonist, and for most purposes, it doesn't really matter. In this case, you can see if we look at the affinity of SIMI36, it has very high affinity, so into the picomolar range affinity for the 5-HT2A receptor. It's also very potent at the 2C receptor, something like a 10 to 15-fold separation. And in principle, that is not enough to get very good separation in an image if these two targets are expressed close to each other. But the lucky thing, or what you want to say, is that the 2A and the 2C receptor are not expressed in the same place in the brain. So even though you get bindings to the 2A receptor, it's somewhere else, so you can actually use this compound to selectively image the 2A receptor. So we then sort of begin to think about, so what about activation of the 2A receptor? Now, can we also make a pharmacological tool that can be used to activate the receptor selectively, and not only to look at the receptor? And that is a game of efficacy. So the ability to activate the receptor, where we measure first in affinity and a Ki value. The other one is typically, we measure that in as an EC50. And also the assays we use to evaluate the compounds, for the ability to do this is very different. So if you then look at the relative efficacy of SIMI36, you can see, well, that is basically equipotent on those two receptors. So if we dose this compound to a human being or something else, we will get an equal activation of those two receptors. So even though it's a compound that is useful for imaging of the 2A receptor, it's not very useful for a selective activation. And if you compare it to something like psilocin and look at the efficacy of that on the two receptors, this is from one particular place in the literature. If you look somewhere else, you get different values. It's important to notice, to stress that, even though the same, if different labs do, in principle, the same assay, they will get different results. So don't, when you see a number, well, I saw a different number somewhere else. Yes, that happens. But here you can see it's roughly a 9-fold separation between the two. And you might say, okay, well, in this case, is then psilocybin and psilocin a much better compound than this to induce selective activation of the 2A receptor? Well, not really. So I stole this slide from a lecture that Brian Roth gave online a couple of years ago. I'm not sure, I don't think they have published it. But what they did here was to look at psilocyin's ability to activate the other serotonergic receptors. I showed you there in the beginning, they have affinity for it. And that's one thing, of course, that is a problem if we're talking about imaging. But as you can see from this overview, it's also a potent agonist of most of the serotonergic receptors. So that also means that when you dose an animal or a human being with something like psilocybin, yes, you do activate the 2A receptor, but you also activate a lot of other receptors. And one of our sort of, ambitions or ideas going behind that, also what you said, Eric, at the beginning, was to, can we sort of, try and come up with compounds that we would be able to selectively activate the 2A receptor to try and figure out, what happens there, and all the other receptors that are being activated by something like LSD or psilocybin, is that also important for their, acute subjective effects, for the long-term acute subjective effects, and also the long-term therapeutic effects and so on. So, and when you move into something like that, so we are potentially talking about a compound, that you need to give that to a live animal, perhaps to a human being in the end, and it has to, go into the brain and do its thing, and then hopefully go away and not do other things. And one of the important things when talking about that is not only respect to selectivity for a compound, does it hit the right receptor, it's also other properties of the molecules. And DMPK is something called the distribution, metabolism, and the pharmacokinetics of a compound. So what happens, where does it go, where does it swim around? And if you read in the literature and also some places on the internet where people, they do recreational use of these, you can find out that so the 2CXs, 2CB and 2CI are known to be active in humans, and again, depending on which compound we're talking about, 10 to 20 mg, whereas the N-bombs, that they also emerged on the recreational scene 15 years ago, perhaps like that. It was reported to be much more, you needed a lot less compound to induce a trip, which makes sense. It's a much more potent compound. But there was also this, it's known to be, this you can drink as an oral solution or take as a tablet, whereas N-bombs are virtually inactive. You do the same thing. So they are inactive after parallel administration. And if you, that's fine, there's nothing peculiar about that. That's an indication of a high first-pass metabolism. It means if you want to compound goes into your stomach, it goes directly through the liver, and that is the way we work, is that we have that cleaning machinery to take away compounds we shouldn't have. And this is sort of an indication, okay, if you have this, you probably have a very high first-pass metabolism, meaning that the compound degrades the compound, inactivates it. And there's a very simple way to get an idea about, is that really a problem or not? We can measure what is called the intrinsic clearance. You use liver microsomes. You take some of the machinery in the liver that chews down compounds, and then you simply expose your compound to that in a tube to see, okay, does your compound survive that, or is it being chewed down? So we compared here 10 different 2CXs and their corresponding N-bombs over here. And if you look at the average of this number, you see you get around 0.5 and you get about 9. And without going into the specifics, the higher this number is, the higher the metabolic liability of the compound is. So when you look at this and do a comparison, then the clear conclusion is that the N-bombs, they are rapid metabolizers. So they're being chewed down very fast, which is also fits with the lack of peroral bioavailability in humans. So as we were thinking about, looking at this compound class to see can we develop useful compounds from this, we also asked ourselves, how is Sympy36 metabolized? Because then we could perhaps do something about that in our optimization of the compounds. And given at the time we were doing PET imaging in pigs, we decided to use pigs as a system to investigate it in there. So we did the incubation both of these liver microsomes and also from liver microsomes from pigs. Did a lot of LCMS analysis to the in vitro, and then we also dosed a pharmacological dose of Sympy36 to a pig, pulled out blood samples, did a lot of analysis, and came up with ideas for, okay, we think it's this or that metabolite, and then we also made these suggested metabolites so we could make a direct comparison so we knew whether or not it was these compounds we were looking at. And basically what we found was this, that initially there's what's called a phase 1 metabolism. What happens is that you oxidatively chew off one of those methyl groups sitting on this one here, so you get to this comp, and then subsequently you put on this large group here. And this is something that, a trick the body uses. If you have a very lipophilic molecule like this, how are we getting rid of foreign objects or foreign compounds, is that we try to make them more water soluble so they are excreted via the urine. And this is exactly what happens with this comp here. You go from something very lipophilic to something that is very water soluble. So there's nothing surprising about this. This is completely standard metabolism of a synabiotic. And we sort of were able to quantify that over time. So we start here, so injected here, you can see initially we have, this is the starting compound, so the compound we dose in, you see that is the biggest peak, and then over time that decays off. And you see this initial phase 1 metabolite, which is formed, we can barely detect it. And then over time, you can see the phase 2 metabolite, so where it sort of ends up is this one here. So that fits very nicely with what you could expect with this. Now, given that we were also working with this in sort of in a carbon-11 labeled version, it was much, it gave us the opportunity to also follow this in live animals and not, having to take our blood samples and do all of that. So this is images from some of the studies that was done during the progression of this compound into humans. So this is scans from a pig. Perhaps you can see it's a pig lying on its right side, so with the paws over like this. So you have here the heads, and this is the lungs and the rest of the body down there. So this is different time points, 0 to 7 minutes, 7 to 20, 20, and so on. And here you can see initially, you see uptake in the brain, which is the images we want to look at, but you can also see large uptake into the lungs. That's something that's often seen. And if my pig anatomy is not completely off, what you can also see down here is you see something in the kidney that over time decays off, and at the end, most of the activity is actually collected in the bladder. We don't know that this is the compound, but it fits nicely with this. So, what about in humans then? Well, this is then a scan for also the full body scan of a human being. Usually you would only look at the pictures up here, but this is then, we assume then that the same metabolism happened in humans. So you have the first primary radiometabolite, so this is chopped off when we get to this primary radiometabolite. And if we just revisit this, what I said, that all of these things that were as a concern, this last one here, no blood-brain barrier penetration of a radioactive metabolite. This is a radioactive metabolite, and this compound in itself is also a super potent 2A agonist, which then gave cause to a little bit of sweaty palms, because when we're imaging this in humans, are we then looking pictures at what this compound is binding to or what this compound is binding to? That complicate matters. But given that there is these three possible positions for radiolabeling this molecule, we then went back and introduced the radio label in a different position, so the position where it is being chewed off. So in this case, the radioactivity does not end up in the rest of the molecule, but in that part that is being chewed off. And if you look over here, then you can see a lot of activity ending up in the bladder, whereas in the other case here, there's no activity. Again, we speculate that this here is the same phase 2 metabolite we proposed in the pigs, whereas this is apparently not formed here in humans. But it turned out that this radioactive metabolite, or this radiometabolite, actually, that we saw some brain uptake with that one, which then confounded the pictures. So it turns out actually the original labeling and all of the clinical data that had been generated to that point, we were safe. There were no problems there. So that's a good thing. Okay. Following that and the knowledge about the metabolism of these compounds, we then embarked on a campaign to try and modify this compound in various ways. And we did a lot of things. It's like playing with Legos. You put on new stuff and take things off, and then you see, does it work or not? And I will not bore you with all those details. You can read it in these papers and others if you're interested. What we came up with the end was we liked these two compounds a little bit more than the others. Call these two names here. And so why did we like them more than others? And well, what you see here is a comparison of the affinities of these two compounds for the 2A receptor and the 2C receptor. That's sort of the two main challenges. These are the ones that are being expressed in the CNS. And for the higher the bars here, the higher the selectivity. And if you compare that with DOI, which is sort of also a compound that's used very widely in preclinical research to investigate the 2A receptor, you can see for both cases, we have a somewhat improved selectivity compared to DOI, both in binding assays and also in functional assays. And also it looks like the one on the right, you see the bars are even higher, so this must be the better compound. This is the more selective compound. Let's advance that one. Well, Adam Halberstadt, he then looked at the head twitch response, and the head twitch response sort of sometimes, that's a behavioral proxy for 2A activation in vivo. And what you can see here is that the top compound here is much more potent and able to doing that, whereas the other one, you need much higher doses and you get a much lower response. But that is also because this is done in vitro. This is just putting the compound into cells and see can it activate the receptor. This is dosing the compound to live animals. Will it go into the brain and do its stuff and all of that? So the in vivo potency of this one is much lower than this one here. And also if you look at this intrinsic clearance, this number I showed you earlier on that tells you something about metabolic liability. The metabolic liability of this compound is much higher than this one. So this presumably is being chewed down to something else in the animal. So even though on paper or in vitro it's a quote unquote better compound, this one actually works better. And we also dose this to live animals, and then you can look at how much of the compound goes into the brain. You can actually monitor that if you dose this to an animal, it goes into the brain and stays in there for long enough to have a pharmacologically relevant response. So Anders, he then subsequently spearheaded a fairly comprehensive investigation of that compounds, also with some international collaborators, to look at this, is this truly a 2A selective agonist? And based on data that I won't dive into here, I think it's not a perfect compound. It does not only hit the 2A receptor. It also interacts with its two cousins, the 2B and 2C, but there is a fair separation. So if you think about your dosing of the compound we do in vivo investigations, we speculate that you should be able to get a selective 2A activation. And others have also picked up on this compound. This is an investigation coming out of the Roth lab. So they managed to get a cryo-EM structure of the 5-HT2A receptor in complex with both LSD and the 25C and NBOH. And won't go into too much details, just to say that there was a lot of surprises. So the pink area is where LSD is binding, and then if you see the yellow part, which is the other one, sort of dives into a completely other part of the receptor, much to, I think, both ours and other surprise. So, but I'll return to this structure in a second. But what they also did a very comprehensive pharmacological evaluation of this compound in their setup, and It's very difficult to boil that down to something, but it looks like this compound does, quote unquote, the same to the 2A receptor as many of the other agonists without hitting other receptors to the same extent. And what kind of investigations can you do with this? Well, this is a paper from Fantagrossi back in 2010 when he then looked at this DOI that we just mentioned. So if you take DOI and increase the dose in rats and you count the head-twitch response of these rats, so again, so a behavioral proxy of 2A activation, then you look at the black ones, you can see at 0.3 and at 1 and at 3 milligrams per kilogram, you get first an increase in the number of head twitches and then you see a reduction. But if you then co-administer this compound here, SB24, that is a 2C antagonist. So in principle, that should block the co-activation of the 2C receptor that DOI induces. And then what you see here is that if you take away the 2C component of DOI, then you see the number of head twitches you're able to induce with that with DOI increases. So Bill, he then concluded here that, well, this sort of indicates that co-activation of the 2C receptor influences the head twitch response. So of course you can argue and say, well, if I stick to a dose of 1 milligram per kilogram, it appears that I'm not also activating the 2C receptor. But also if you go and look at sort of the pharmacological data of DOI, you probably do, but it's something that doesn't read out in that assay. So Halberstadt has looked at 25-CNBOH as many others and done this, increasing doses of this compound will lead to an increasing number of head twitches. And the question is, and once you begin to get up in very high doses, you see this sort of, bell-shaped curve in the top here, which, indicates, could we also at these high doses begin to see 2C activation? That's possible. But the question is, could we then pick another dose where we get a significant activation, or we get a significant induction of the head twitch response, and if we then stay at that dose, not touch other receptors? So he basically did a similar investigation here as Fantagrossi did, that with the same dose of 25-CNBOH, well, if you just have vehicles or just dose this with saline, you do see the induction of head twitches. If you co-inject an 5-HT2A antagonist, you shut it down. And if you take the same 2C antagonist as over here, you see it has no effects on the induction of the head twitch response. So basically the same scenario as you have here with 1 milligram, that at least at this dose, if we believe this setup here, we do not get subcellular additional 2C activation. So we are curious to investigate also all of this clinical data coming out that it appears that with a single dose of a psychedelic, you get this rapid onset and also persistent effects of psychedelics. So we reached out to Charles Nichols, and he had published this very interesting model where he was able to replicate what appears to be this single dose persistent antidepressant effect in rodents. So very briefly, what he showed that, so if you take rats and if you dose them with either psilocybin or ketamine, and then basically leave them alone for a week, and then test them in the forced swim test, and then if you compare the white bars here, most of these animals are just completely immobile, they're not swimming or climbing, whereas those that receive ketamine and psilocybin, they are active, they're swimming, and that is seen as sort of an antidepressant effect. And if you then take the same animals on day 35, you can see those that receive ketamine are now the same as saline, so the quote-unquote antidepressant effect has worn off, and that's the same you see in clinical setting with humans, that yes, ketamine can have rapid induction of antidepressant effect, but those effects also wears off much faster than with psilocybin, where in this case here, we still see this, again, single dose after 35 days, you see this increase in swimming, so interpreted as an antidepressant effect. And going back to this, this is psilocybin, so which of those receptors that psilocybin activate is responsible for this effect? So we reached out to him and asked him, would you be interested in investigating 25-CNBOH, and he agreed to do that. So he did it in his setup with his Vista Kyoto rats, dosed them at the first day, and then basically leave the animals alone. And after 35 days, then test them in the forced swim test. So that's the identical setup that you just showed. But then he also, he kept the animals up to week 12, so after 84 days after a single dose, basically just letting the animals run around in their cages, and then score them at these 2 dates. So you can see this is then saline, so this is immobility. And you can see as the animals grow older, they get a little bit more immobile, and I guess that's okay. And if you then look at the cohort that receives a single dose of psilocybin, you still see, so after 5 weeks, that's basically the same setup that I just showed you. But he was able to show that also this effect stays on for another additional long period of time. So after 12 weeks, you still get this antidepressant-like effect. So we were, of course, curious to see what happens with the animals that receive the 25-cNMH, and basically we see roughly the same. So we get the same in this assay persistent antidepressant effect following a single dose. And one can, of course, begin to speculate a lot about this. Does this then mean that the antidepressant effects of psilocybin, or those that look very promising, are they also due to the activation of the 2A receptor? Well, we would think so, and it's also spurred us on to continue working within this area. So we initially published this compound, I think was 10 years ago, and then subsequently other labs have also begun to pick it up, and when we get requests, we're happy to share the compound for those who want to investigate it. It's also commercially available. There's no strings attached or anything. It's just like any other compound you can buy and use in your investigations. What it's not so useful for is that something with imaging, because we've been in the imaging area, so we also look at the tritiated version of this compound and also the carbon labeled version, trying to do some autoradiography, and we also looked at Anders looked at the specific binding here, but the take-home message is that it's not working very well. And also we looked at the carbon labeled version. This is some of these pictures, where we zoom into the pig brain, and this is the carbon labeled version of this compound. And if you compare to a similar image with SIMP36, so this is again, it's a pig brain just seen from the side on. So what you would like to see is what you see with SIMP36. You have higher uptake in cortex and no uptake in cerebellum and the other areas. And here we actually see, a very mixed bag of results. I should say that this is based on a single experiment, n equals 1. So if one were to do it again, it might look different. But there were also some other things about this that led us to conclude that let's not pursue this anymore. So just to sum up what the difference between these two compounds, the SIMP36, that's useful for selective imaging of the 2A receptor if you want to do those kind of experiments. But given its equipotence on 2A and 2C, it's not very useful for the selective activation of that one. But on the other hand, if that is what you want to do, then this compound is a useful tool for you to use in preclinical research. It works well in animals, and of course you can also use it on correlates and so on. So useful for selective activation, but as I also just showed, not for selective imaging. So depending on the kind of investigation you want to do, pick your favorite. Now, over the years, we spent a lot of time playing with these molecules, trying to understand it more and develop a pretty good synthesis so we can actually make substantial amounts of this compound. And one of the things we also begin to see, have also been interesting, of course, is trying to understand its interaction with the target, with the 5-HT2A receptor. And one of the things that makes this very complicated is that this is a very flexible molecule. And what do I mean by that? Well, it can adopt very many conformations. So the bonds that I've mixed here with the yellow arrows, if you compare that to say that you take two frying pans and you tie them together with a string, it becomes very floppy. And that is indeed the case. This is a very floppy molecule, so it can adopt multiple different conformations. And which of those are then relevant for the interaction with the 2A receptor? We didn't know at the time. But we were, of course, curious to find out. And we're not the only ones curious about that, because Nichols, he was, of course, extremely active in this area at that time, and sort of he did this very nice study where he basically did that investigation on this compound, trying to think about, okay, what about if we look at the different conformations of this one and try and lock that up? So he came up with this compound, and it's actually a very interesting compound, but I don't think a lot of people have heard about it, and it hasn't been used in a lot of studies. And I think the main reason is that this is quite tricky to make. This is not something you make 100 grams of and use for different things. It's very tricky to make. But at that time, when this was published and we began to think about that, wouldn't it be cool to see if we can sort of make a hybrid between these two structures? So if this is, without this part here is our best bet for a selective 2A agonist, and this sort of traps the N-bombs in the bioactive conformation, then this must be the best of two worlds. So we had been working on that for some time, but the chemistry is very tricky. So the synthesis of these compounds is by no means straightforward. And then when this cryo-EM structure that I mentioned just previously came out, and we began to look into that and begin to compare that with up here, and if you then zoom in, and so in here in gray, so that is actually the compound sitting in there. And Is then this the bioactive conformation of 25-CN-NBOH? We don't really know, but it's probably the best bet. So this is a cryo-EM structure came out where it was able to string in there. So could we make a compound where we then lock our compound here in the same conformation as in there? So we did that. So this is these 4 compounds. There are 4 stereocenters, so 2 stereocenters, so that gives in all 4 possible compounds. And this is an attempt to try and sort of rationalize what came out of it. So the structure you see in gray and red here, that is in all of these 4 ones here is the same one. And that is the conformation of 25-CN-NBOH in the cryo-EM structure. And then here what you see in the orange and the blue and the green and whatever that is these 4 compounds here, energy minimized, we then map on the structure there. And this one looks really horrible. I think you can agree with me. These 2 here, and there's also something wrong here, but that is about this cyano group. Where does it end up? Is it sitting up here or is it sitting down there? So it's really, really tricky to fit these 2. Whereas in this case up here, that's reasonable. You can get a reasonable fit. So we were initially thinking, okay, so hopefully this will then be the perfect molecule. This is really great. So Emil and Jitke spent a lot of time and others prior to them making these compounds. We gave them to Anders and got back to pharmacology. And well, compared to 25-CN-NBOH, which that's a single-digit nanomolar agonist, well, we see a large drop in affinity or in efficacy, both on 2A and on 2C. So that was, of course, extremely disappointing, and also the other compounds were, barely 2A agonists. That was perhaps not so surprising, but we, I spent a lot of time and effort putting it into this one, and it was, of course, extremely disappointing. And then one could ask, what is the reason for that? Well, it could be simply that this compound doesn't map the right conformation. You can also see we introduce some extra bulk, so this top part here, perhaps there's no room for that. Looking at the structure, there should be it, but no. Okay. As you probably also know, if you've been following the field, that as these N-bombs came out, both as Nicholas initially published them after Alfheim had been working on them, also sort of the recreational use on this began sort of to spread out, and they were easily available. And there's a couple of reviews or a number of reviews that sort of summarize some of these things that say that the N-bomb drugs are highly toxic and their intake has been associated with severe adverse reactions, including death, and the only safe use of this one. This is very harsh statements, and it is hard statements because there are severe problems. And one of our ambitions was, could we try and develop a selective 2A agonist we could actually take into humans and try and do the same kind of investigations? What happens if you selectively activate the 2A receptor in humans? And the more we had been working on these N-bomb compounds over time, we were beginning to lose faith in that it would actually be able to do that. Is that actually the right compound class to work on? So if the N-bombs are toxic, does that mean that we shouldn't, Gitta and her crew should stop doing this? No, but you have to remember that when you do these kind of investigations, you do that with very, very small amounts of this, and this is tracer doses. So the people receiving these PET scans, there is absolutely no pharmacological effect from this one because the dose is so low. There's no pharmacological effect and there's no toxic effect, so there's no concern there. So at that time, or some time ago, we then decided to go back and say, okay, so the N-bombs were sort of derived from the corresponding 2CXs, and there was a lot of hype and saying, no, this is so much better, they're so much more potent and so on, but are they really that much better? If you look at 2CB and look at its potency at 2A receptors, it's in both for 2A and 2C, so it's single-digit nanomolar, so this is very potent compounds, and of course when you then look at the corresponding data for the N-bombs, yeah, they are more potent, but they are not much more potent, they're a little more potent. And also if you compare with something like psilocin, well, that's roughly the same as 2CB. Now, if you sort of compare compounds, not necessarily for one target, but what is a good compound irrespective of what target it's hitting? There's a bunch of different metrics you can use to compare them. One of the very simple ones that also intuitively makes sense is something called the ligand efficiency. So that means something when you then look at the energy of binding compared to N, which is the number of heavy atoms. So that basically just means how big is your molecule, how many pieces do you need for it to do what it's supposed to do. And if you count the number of heavy atoms in 2CB, so that's the oxygen and the carbons and the bromine and nitrogen, there's 14 in all. So that's the number of Lego bricks you need for it to activate potently at the 2A receptor. The same number for in this case, it's 25%, so we've added on bulk, we've used more Lego bricks without really gaining too much here. And if you look at something like psilocybin, it's the same number of heavy atoms, and you can see you get the similar potency. And if you do the math here, then you end up with numbers. And again, please don't write them down. This is just an indication that the higher the number is, the more efficient is the compound. So it's not a very efficient compound here. We can easily get high activation of the 2A receptor without adding this bulk over here. And again, going back to the metabolism, one of the problems with this N-bomb class, I think it's fairly well established now, is that you make the compounds much more greasy. They get much more prone to metabolism, which also can give some issues with tox. So the N-bombs, this particular one here, is super useful as a PET ligand, but it may not be very useful to be used as a pharmacological tool. So what we did was to begin to think about, can we not do something with this one? So in the same way that these N-bombs are flexible molecules, the same can go for the 2CXs, they are also flexible molecules. So could we go back and play with this and perhaps do some things, again try to trap these compounds in their bioactive conformation in the same sense that we have tried, and others of course have tried for the N-bombs. So what about making conformationally restricted analogs of 2CB? Well, again, we're walking in the footsteps of giants. So Nichols, of course, have looked at this and with compounds like 2CB, TCB2, and this one over here, where he's done exactly that, was to try, let's take this part of the molecule and make it less flexible so we can lock it in a particular conformation so it has the possibility to interact with the 2A receptor. Again, these perhaps have not been used widely. Again, the synthesis of these are tricky. We've made these, and this is not simple. So we thought, okay, can we do something similar and also, make the compounds more readily available and perhaps achieve selectivity? For even though these compounds are super potent 2A agonists, they're still not selective for 2A. So we thought, let's try this simple trick in doing something similar where we build rings, reduce flexibility. Let's introduce a 4, 5, and a 6-member rings on these compounds and see what happens. So we did that. So if you compare that with 2CB, so what you see here is just simply the potency on 2A and 2C, and if you then close it into the 4-membered ring, you basically get a compound with very similar profile. It's a very potent 2A agonist, but also a very potent 2C agonist. So interesting compound, but not very useful. We have many of those. Then look at the 5-member ring. See, up here there's a chiral center. That means there are two mirror images of this compound, a right hand and a left hand. So there are also pharmacology for both of these. So we separate these and check the pharmacology, and we got two curves that look like this. And again, this was sort of, well, we have many compounds that look like this, perhaps not super interesting. And then we did it for the same thing for the 6-membered ring and did the testing and got this. And that was a little bit more interesting, because what you see on the right up here is that for 2C, for this compound here, it is no longer able to activate the 2C receptor. It's not a very good 2A agonist either, but we have completely suppressed its ability to activate the 2C receptor. And ironically enough, this compound was reported by Nichols some time ago as an intermediate in one of these compounds where they tried to lock in the conformation of the N-bombs. But for some reason, they didn't look at the pharmacology, or perhaps they did and found it uninteresting. But important also to mention, this compound was actually known when we did the investigation. And if we take a closer look, it is just the same data. I'll show you one more time. So on 2A, it's I don't know what you say, it's a 70 nanomolar on 2A. And if you then also test this in antagonist mode, so not its ability to activate the 2C receptor, but can it block serotonin's ability to activate it, it's then an antagonist with an IC50 around 640 nanomolar. So we took this sort of as a starting point and did a lot of optimization, made a lot of compounds. Again, I won't bore you with the details, but just say that we came up with this one, which ended up having an interesting profile. So the one enantiomer that looks like this was again perhaps not very interesting and very similar to this one up here. We've simply exchanged the bromine with the trifluoromethyl. But we then look at the same enantiomer, or the same, this one here. Well, we saw that when you do the switch from the bromine to this case, the trifluoromethyl, we see a boost in its ability to activate the 2A receptor, while it maintains its inability to activate the 2C receptor. And if you do the numbers, then you can see we increase the potency from around 70 to around 3 nanomolars. And if you then look at again, Anders tested these as antagonists in antagonist mode on 2C, then we then get an IC50 around 300 nanomolar. So it gets more potent on 2C, and it becomes a more potent antagonist in this assay of 2C as well, but we get a decent separation. And again, we have a compound that we then believe could be used to selectively activate the 2A receptor. We did a lot of modifications on this, is just to show that when we give the stereochemistry, we got the X-ray structures of the least potent enantiomer that showed us this was the one, so we conclude this was the most potent one, and we also have subsequent X-ray structures that confirms this. So we are very confident about the absolute configuration here. So just to finish up here, just did a comparison with some of the other compounds that are out there. 2C-TFM, one of the most potent 2A agonists ever reported, again by Nichols. So this again dips into the picomolar range on 2A. And what happens then if we tie this extra ring on that I just showed to you? You can see that you lose some potency on 2A, factor of 4 or something like that, which you could say is a step in the wrong direction. Yeah, but it's still a very potent 2A agonist, and we completely suppress its ability, at least in this assay, the compound's ability to activate the 2C receptor. So we are willing to give up on some potency on 2A if we can get selectivity. And then to the point of this atom economy or ligand efficiency. So is there anything in this ligand we don't need? Can we chop further things away? So we did that sort of investigate if we make some small modifications to the compound, what does it do to the compound? Just add a simple methyl group out here. Well, it's not good for potency. If you take away one of the methoxy groups, you can see EC50 goes up quite a bit. The other methoxy group here, well, you still have a fairly potent agonist, again, but something like a factor of from 3.2 up to this one here, so not a step in the right direction. And if you remove the trifluoromethyl group and get to this one, that compound is not a 2A agonist. So we sort of conclude that we need everything what we have in there, and we sort of decided, let's see if we can take this compound into humans. So we established a company some years ago, renamed this compound to LPH5, and have now been pursuing this compound for some time. There's a preprinter out there where we have done a thorough pharmacological characterization on this on a number of different receptors, and you can see this is data from 2C. Now it's not an antagonist, but it's a low potent agonist, and in other assays as well. I won't dive into the details, you can have a look at that one. So what about in vivo pharmacology? So we looked at this in the in vivo, in the head-touch response. Bettina in Aarhus did that with many different doses. I'm just showing you here that it's just cumulative data on head-touch response. So you see the onset of the head-touch response and then dies off after something like 1.5 hours. And if you go to a lower dose, well, you barely see any reduction of the head-touch response. And we again reached out to Charles Nichols if he'd be willing to check in this single dose persistent antidepressant effect model. And he did so and got basically the same results that we saw with 25C and NVOH. This is, again, the data from e-mobility in swap swim test, 35 days after a single dosing. And in both cases, both a 0.3 milligram and a 1.5, so a dose where we barely expect to see the induction of the head-touch response, we get a persistent antidepressant effect in this model. And that's also reassuring to see that there's more and more data coming out for this model from different chemotypes, but you could argue that this was just the effect of activation of some freak receptor that we don't know of. But now there's more and more different 2A agonists that show the same profile, which again gives further indication that also the therapeutic effects of psychedelics is probably 2A mediated. So we are now currently advancing this compound into clinical trials, and what do you need to do that? Well, you need to be able to protect your compounds. So we got our patents. We made a lot of the compounds, more than 6 kilograms in total. We completed toxicology studies in order to qualify that from first in man, and about a month ago, we submitted a clinical trial authorization application to the French authorities, and we're currently waiting for them to come back to us, and then if everything falls into place, we should be able to do first in man studies with this compound, hopefully at the end of this year. So my time was up 10 minutes ago. So what I tried to show you was, all compounds are not born equal. There's a difference between the different applications of the compound you need to take into consideration when you develop these. Is it for imaging? Is it for preclinical studies with the selective activation of the 2A, or do you have ambitions of actually taking these compounds into the clinic? Brings me to the final and most important slide, of course, the acknowledgements. What I've showed you here today is the contribution of a great many people over the years. So from respect to the chemistry, it's Sebastian, Martin, Emil, Ida, and Jens that helped us recently. All of the metabolism studies and all of the analytical works that were done there was by Charlotte and Andreas. A lot of in vivo pharmacology, from Anders, Hans, John, and David that helped us with this. And a lot of the in vivo pharmacology I didn't show you also here. Bettina and Bill, Adam, Charles, and Jesper from the university helped us out. All of the imaging work and all the radiochemistry was part of the SIMBI project. And of course, I also thank those that sponsored our research over the years. Thank you.